Welcome back for Learning in Genetics Part 2. I'm going to switch over to my PowerPoint. And I think we're right about here. Genetics and human speech and language. There are lots and lots of articles about this, obviously. But we've just been talking about how the gene FOXP2 seems to be important in the development and learning of birdsong in zebra finches, and it's been fairly well studied in zebra finches. What about humans and learning language? Is there a genetic component that would be similar to, say, zebrafinch song learning? Well, language is definitely unique to humans. Usually by the time, at least the complexity of language that we use is. Usually by the time a person is 3 or 4, she knows thousands of words and can construct complicated, meaningful sentences that easily tell us, what she's thinking, right? Aspects of this ability to learn language are encoded in our genetic makeup, seem to have an innate component. We have a genetic predisposition to learn language. We have a language instinct. We have a sensitive period in which we need to learn language in order to learn it well. Researchers have shown in a few cases where human children are not exposed to language, they struggle to develop meaningful language as adults when they are, or as older children when they are found. So there's this critical or sensitive period of time. I also noticed, anyway. So is there, are there similar neural structures in humans compared to, say, zebra finches? Are there genes in humans that contribute to learning language, similarly to what we see in zebra finches? Well, I would say, if we're going to study the genetic component of learning language, we're going to have to look at people who have disorders or deficits of speech and language, right? We might look for people who have difficulty in a whole bunch of different categories of speech and language. There are lots of types of disorders, and I'm not a speech and language person, so I'll just give you a couple of examples. Like, you could have someone who has articulation errors, or has trouble with the production of sound, or has trouble with grammar, or expressive language, or is unable to communicate in writing as well as orally, word repetition. There's a lot of different kinds of deficits in speech and language, right? And it's hard to categorize them, if you will. It turns out that a lot of these disorders are highly heritable. They run in families. We see families that have individuals who have very similar types of speech and language disorders. There are twin studies that suggest very high heritability of certain kinds of disorders. So a lot of research has been done on that. Well, how do you go about finding the genetic basis for a speech and language disorder, right? Well, there are several different approaches that one can take. One of these is called the candidate gene approach. So you have to use pre-existing information about how the disease occurs to hunt for genes that are good candidates to study. Well, we could possibly look for like the FOXP2 gene, because we know it is so important in song learning in finches. But for the most part, we have very little knowledge about the mechanisms of speech and learning disorders. So this strategy isn't super useful. Instead, researchers have used what's called the positional candidate approach. So the idea here is to track the inheritance of markers, genetic markers called polymorphic genetic markers, in families. And in families that display, at least some individuals display speech and language disorders. So you have some individuals in the family who have normal language and speech skills, and some that have disorders. And what we're looking for then are certain, looking at these polymorphic genetic markers. They're hunting through the whole genome, looking for regions of the genome, specific regions on different chromosomes that stand out as being different in people who don't have the disorder versus people who do have the disorder. And then, once you found we found a region over here on chromosome 5 or on chromosome 7, then you start, once you find an interesting region on, say, chromosome 7, then you start to study the genes in that region and say, are there any particular genes that may have functions of interest? So we know that individuals who have the speech and language disorder have this interesting marker, genetic marker on chromosome 16 or whatever. Now we're going to look in that region for genes that might be involved in language development, for example. Okay. And so along comes a family known to us as the KE family. This was really fortuitous because this family is a 3-generation family. The pedigree here is kind of funky looking. This is a pedigree of a family. The squares represent males, the circles represent females, and males and females that are mated. If you can see, if you've never seen one of these pedigrees before, this would be kind of the grandparents here of this whole family, and they mated and had these children here. Okay. And then it looks like that male died, and then the mom mated again and had a whole nother set of children. Okay, and again, etc. Okay, so what researchers found with this family is about half of the individuals in the family had a significant speech and language disorder. Those individuals are marked here with dark squares or circles. And they didn't really have any disorders related to their IQ, but they had major speech and language disorders, and they all were kind of similar. And looking at the pedigree, researchers kind of hypothesized that there's a single gene that's contributing to this speech and language disorder, and it seems to be dominant, which means if you get one copy of this mutated gene, you show the disorder. All right, so now they think they've got a family in which you've got a speech and language disorder among a huge number of people. It seems to follow that there's a single gene mutation contributing to this disorder. Now we just have to find out which gene is it. They begin the search for the mutated gene in the affected members of this KE family. Okay. Well, before we move on, maybe I should answer question 2 here. I kind of forgot about that. So question 2 says, what kinds of speech and language disorders or deficits do the members of this family have? And you can look this up if you want to online as well, but they have articulation errors. They have difficulty coordinating their mouth movements for making speech. They have language impairments in oral and written expressive language. So if you ask someone to write down as many words starting with the letter D as they can think of, this was really hard for people who have the disorder. They had very few other cognitive deficits. So it was primarily in speech and language. All right. Did they find any neurological abnormalities in the KE family? Yes, they did. They found some structural or functional abnormalities in the basal ganglia of the brain. Right? The basal ganglia plays a role in speech and language. That's no surprise. But they found some very specific differences between people who have the disorder and who don't in this region of the brain. Okay, so then they begin searching for the mutated gene in these members of the family. So they started hunting around. They were using what we called the positional candidate approach to finding a region of the genome that looked interesting. So they used polymorphic genetic markers they hunted for them that are the same in individuals who are affected, and they found one region on chromosome 7 that looked pretty good. So chromosomes are made up of typically made up of 2 arms. Each chromosome has like a long arm and a short arm. One is called the P and one is called the Q arm. And then each chromosome is broken down farther into segments. So they found a region on chromosome 7 called the 7q31 region. All right? They even named it. Let's call it the speech 1 region. Turns out there's like 100 genes in this region. And it seems to be the area where that kind of is similar among all individuals who have this disorder. So they're looking around at these 100 genes and they're like, oh, which one is it? Which one is the mutated one? And they catch a lucky break because in another unrelated individual, we call that individual CS, this individual had a very similar speech and language problem to the members of the KE family. And this individual had kind of a very big chromosomal mutation in which chromosomes 5 and 7. So everyone has 2 sets of chromosomes, right? And you have 23 pairs of chromosomes. You have a pair of chromosome number 1, a pair of chromosome number 2, a pair of chromosome number 3, etc., right? Well, one of the, in this person named CS, one of the chromosome number 7, so let's just say these are chromosome number 7s, and one of the chromosome number 5s kind of had a mix-up. Like a piece of chromosome 7 got swapped with a piece of chromosome 5. So the person still had all of their genetic makeup. There was just kind of a swap, okay? That's called a translocation. In any case, what's interesting about this is the break, where the break happened in chromosome number 7. So this is chromosome number 7, if you will, and these are chromosome 5. Where the break happened was in a particular gene, and that gene was called FOXP2. Okay. The patient CS has a disrupted or mutated copy of one of the FOXP2 genes in the chromosome that has this translocation on it. So maybe it's FOXP2 that's also affected in the KE family. What about their family? Is the mutation that seems to cause this speech and language disorder in the FOXP2 gene? Researchers look, and guess what they find? It is. All right? It turns out that there's a single nucleotide mutation. So if you look at the whole gene, only one nucleotide is different, but a mutation nonetheless. That leads to a change in the protein that's made, and we think then a change in the way that individual develops. And it turns out that individuals with just one mutated copy of the FOXP2 gene, so here's our FOXP2 gene there on chromosome 7, 7q, one mutated copy of this gene, people with one mutated copy start to show the speech and language disorders. It's a dominant mutation, if we want to say it that way, okay? Okay, so, well, what does the FOXP2 gene do? Right? That's the next question. And remember, this research is going on at the same time as research on the zebrafinch. So, we learned quickly that the FOXP2 gene is what we call a forkhead box gene. It's in that forkhead box family of genes, and these genes are transcription factors. That is, they code for transcription factors. Transcription factors are proteins that can switch other genes on and off, can turn on and turn off other genes in the cell. And they seem to be very important in humans in cell specialization and pattern formation during development. So we've got this important gene because it produces a protein that regulates other genes. It turns other genes on, it turns other genes off, and it tends to happen during development. What kind of genes does FOXP2 target? a lot. That's all we know. We don't know everything about this particular gene, but presumably, whatever genes the FOXP2 protein targets seems to affect the development of the brain structures that are known to be impaired in people in that KE family that have the speech and language disorder. So right now, this is where you could type in like, all right, this is where we're going to have to do more research here to learn more about exactly how this FOXP2 gene works. Which genes does it target? How do those turning on or turning off those genes influence the development of this region of the brain, right? The basal ganglia that is involved in the development of language somehow. That's the fun of it, right? But here we've got a strong candidate gene for whose protein product contributes to the development of language in humans. Do individuals with more common types of speech and language impairments show mutations in this FOXP2 gene? Researchers have suggested no. What that probably means is there are multiple genes that are involved in the development of speech and language learning. Absolutely. That wouldn't be a surprise, would it? Are there other genes that contribute? Yes. In fact, researchers have found in the last 15 years many, like a dozen more genes that seem to be contributing to speech and language impairments, or whose products, when they're mutated, seem to contribute to speech and language impairments. Interesting. And then other researchers are like, oh, speech and language is such an important part of modern humans. It'd be really interesting to take a look at the FOXP2 gene and see if, like, our FOXP2 gene is different from, our closest relatives, and when did our FOXP2 gene mutate, and did that have an effect on our ability to learn and use language? Wouldn't that be cool? You could find, this moment in time, when the FOXP2 gene mutation occurred, that gave us, instead of our chimp ancestors or our other relatives, better ability to use language or something, right? So it's super interesting. How does FOXP2 relate to the evolutionary origins of speech and language? Well, researchers have found a few things that are really interesting. One of them is that it's highly conserved among species. So we see FOXP2 in fish and in reptiles and birds. Birds are a type of reptile, right? And in mammals of all kinds, and it's almost the same in all of these animals, but there are some differences. And one thing that's interesting is the FOXP2 protein in chimps and humans is different by 2 amino acids. In humans and mice, it's only different by 3 amino acids, but researchers are like, oh, chimps have a different FOXP2 protein. I wonder if that's why humans have such good language skills, because they have a different version of FOXP2. Maybe a mutation happened that enabled us to develop language differently than, say, chimps. And initially, researchers started to date when these mutations could have happened, and they believed that these 2 human mutations happened about 200,000 years ago, just about the time in which modern humans are evolving or emerging. And so they're pumped up, and they're like, yeah, maybe these 2 mutations gave our human ancestors such an advantage that the mutations were highly selected for and just swept through the whole human population, and that's why modern humans have such good speech and language skills, because of this mutation, or these mutations. Well, more recent research has suggested that both of these mutations probably occurred way farther back in time. way millions of years back in time. And so chances are that these mutations were not suddenly selected for 200,000 years ago, but were with us as we diverged from ancestors that also gave rise to chimps. Okay, so that means other members of the Homo genus, and like Neanderthals, have the same FOXP2 gene that we do, right? That's an interesting story, and it's certainly worth evaluating. But I think what the most important point of this lecture is that when we take something like language or bird song, we tend to think that language must be learned, right? Language is something that we learn, and it's true. It is. We tend to think of that as being nurture, part of our nurture. We learn Dutch if we grow up in the Netherlands. We learn American English if we grow up in the United States, etc., right? We learn Amharic if we grow up in Ethiopia. It's very much a learned or nurture component. And yet we also now see that our genetic makeup contributes to the ability to learn these languages. FOXP2 is definitely one of the genes that contributes to our ability to develop speech and language. So here we've got, again, this really interesting relationship between nature and nurture, if you will, between genes and learning in this case. And I think that's pretty interesting. Now, language is really important in communication and in our ability as humans to develop culture, for example. And so we'll talk again about language in future lectures. But for now, that's where I'm going to leave you. All right, I think that's the end of this lecture. Thank you very much.